So what we are trying to do in this project is to use or to mimic the way in which cells synthesize their own components to develop new synthetic methodologies that are more efficient, that produce less waste, and therefore that are more environmentally friendly. So we are trying to use the compartmentalization strategy of cells, for example, and we are trying to regulate the chemical synthesis that are needed in the chemical industry in a metabolic way.
Well, can peptides serve as catalysts? Peptides are the small versions of enzymes. They have the same building blocks as enzymes, but they're way smaller, 3 amino acids compared to 300 or thousands of amino acids. And I'm really intrigued by how catalysis has evolved and how one can make catalysis more efficient. So a theme that really fits very well into Classy that has been going on in my lab for a long time is to ask, by asking whether peptides can be catalysts, which role have they played in the evolution of catalysis, and at the same time, how efficient can catalysts based on peptides be?
In the main part of our work is a replication process, or several replication processes that we study. All of them are initiated by a template molecule that can associate with shorter substrate, which can react and give rise to this replication process.
It's a Fed Open project and it will last for 4 years, and the idea is that the project is really innovative. It uses basic science but to bring forward a really breakthrough idea. And what the Classy project is trying to do, as Andres also explained, is to create a microreactor that can simulate cell-like activity and catalysis in reactions. And this would be really great because it can cut down on lots of waste products that are usually generated when synthetic reactions occur using many more processes than what instead Classy is aiming to reduce and hence limit the waste outcome.
This is a very challenging project. I mean, it's easy to say, but it will for sure difficult to do. And for that, we have really worldwide experts in the areas that I mentioned. We have, for example, Gonen Ashkenazi, who is a real expert in peptide self-assembly and peptide replication, and he will make the molecular effectors that will regulate this type of reactors. We have then 2 experts in microfluidic technology. One is academic, the group of Wilhelm Hack from the Radboud University of Nijmegen in the Netherlands. And we have also a company which will be able to exploit the reactors that we will produce, which is Micronit. And then we have 2 experts in biocatalysis, one with peptides like Helma Wennemers, and also from Helma Wennemers is from ETH here in Zurich, and also Wolfgang Krautil from the University of Graz, expert in biocatalysis with enzymes, and myself, who will be responsible, let's say, for the building blocks that will allow programmability of the catalyst that we are trying to make.
My group is located at the University of Graz in Austria. Our focus is biocatalytic synthesis, meaning that we use biocatalysts, enzymes, in organic synthesis. We try by using biocatalysts to shortcut synthetic routes to biologically active molecules, for instance, or natural products. And we achieve that we circumvent protection strategies and exploit the rather specific reactions of nature.
I am originally from Serbia, and I'm a chemist by education, so I completed my bachelor's in chemistry at the University of Belgrade. Okay, and my direction was basically organic chemistry, or yeah, that's the field that I saw myself in, and for my master's I moved to Spain, to ISIC, to do organelle catalysis. So I spent a year there. Afterwards I came back to Serbia for a little while and I started I took my first steps in biocatalysis, and that's where I saw like a whole range of opportunities that I wasn't aware of previously. So I decided to take advantage of that and take it to a higher level. So I decided to start a new PhD in biocatalysis at the University of Graz in Austria.
I've been working at Micronic for 15 years almost. So I've been asked to join the Class C project, where we are developing the microfluidic chip. So we're taking the chemistry that's developed by the other partners in the project and put it onto a microfluidic device. So to extend the possibilities of the chemistry. So our aim is there to make small droplets and put the chemicals inside the droplets and let them react to create new reactions. So it's for us a completely new type of product. It's very exciting for us to develop something completely new.
I'm Jeanette. I'm from a company called Accelopment based in Zurich, Switzerland. I'm the administrative project manager of the CLASI project, the FET Open project funded by the European Commission under Horizon 2020. And our role in this project is twofold. On one side, as I said, I'm the administrative project manager, but we also have Emily involved, who will be supporting the project on the communication dissemination side. We're very, very pleased to be involved in this project as a project partner, as a small company with fantastic scientists from ETH, the University of Madrid, from Israel and the Netherlands. And systems chemistry seems to offer huge opportunities that we would like to see go forward. And we're looking very much forward to working with all these scientists, PhDs, postdocs, senior scientists over the next 4 years, and hopefully get some very interesting results that can be taken further into future projects that we can be involved in.
I just started my PhD in Radboud University in Wilhelm Haug's group, and I will be working on enzyme encapsulation in beads and compartmentalization of reaction networks there.
I recently started as a postdoc in the group of Professor Wilhelm Haug, and the Haug group is interested or keen on understanding life and basically also how everything started. So my background, I studied at the University of Münster, where I did my postdoc with Frank Glorious in the development of, I would say, synthetic organic methodology. And I was looking for new adventures and new challenges, and this is why I then joined the Haug Lab. And I'm really looking forward to also participate in the CLASI actions in the end, because I think the goals are highly visionary, and I really enjoy working in an interdisciplinary environment. So yeah, I'm looking forward to that actually. Yeah.
Well, there's a lot of complementary expertise coming together. We have someone who really knows how enzymes work and how to evolve enzymes. We have, on the other hand, someone who really knows how to compartmentalize, and that is important for chemical evolution. And we have people who are really experts in supramolecular chemistry, and with us as experts in catalysis and supramolecular chemistry on top, we feel very happy and it's challenging, but we'll certainly get something interesting out. And then it's research. You never know where it takes you. It's entering the unknown, so you never know what's going to come.
I think one of the challenges is the consortium is quite interdisciplinary, so we need to find out where we can benefit from each other, and I think it would be quite important to have good communication between all of the different groups.
The end vision of having like a dynamic cell-like system is quite like almost science fiction-like, but definitely something to strive for. And I mean, having an inspiring goal is definitely something that motivates work. So yeah, I definitely agree with the statement that it's better to aim high and miss than to aim low and hit. So yeah, I think it should be like that end goal should be enough motivation to get the project going.
From a collaborative administrative perspective, we feel that classes should be very much go straight forward, because some partners have been collaborating before. So after the project meeting, after the kick-off meeting, I'm highly optimistic that things will go smoothly and work out very well in Classy.